thinking to briefly pick your brain about a related but different project I'm doing. So one of the things we're playing around with in our Rejuve project on the research side is just using AI to help us suck in as many biomedical data sets as possible from the web into like a common integrated knowledge graph. And this gets back to the strengths and weaknesses of large language models that we were discussing before, actually. So for actually finding patterns among a large number of integrated data sets and understanding sort of what's causal or what's correlative in these data sets. I mean, LLMs, transformer neural nets, are not that useful, and we have other AI methods that are more useful. On the other hand, we're finding LLMs are useful for automatically interpreting the metadata in data sets. So it's like you have a research paper someone published, there's some spreadsheet they put online, or it's tab separated data file which has the data in it. Usually the row and column headers are some incomprehensible abbreviations, and we can use the LLM to figure out these incomprehensible looking abbreviations, figure out what are the row and column headers in the data set someone put online, and then as a result you can automatically suck in a huge amount of online data sets in a way that makes explicit the linkage between them, right? So it's understanding the metadata of the data set. So in this research, then we're constructing a big like metadata set of all the publicly available biological and medical data sets. Now that's I mean, it only covers what's been studied and it covers what's been put online openly, but that's in some domains it's a decent amount. Like there's a lot of gene expression studies of different tissues in people under different conditions. I mean, pretty much every study that's funded by the US NIH has a rule they have to open up the data when they publish their research. Now, if they can make an excuse like this might compromise someone's privacy, then they only quasi open it up, and you have to be someone that they like to get access to it. So a lot of DNA data you can't actually get unless you work at some major university or something. But you can get a lot of clinical data studies. You can get gene expression studies. You get studies across organisms. So that, I mean, one thing we're going to do with this is just make a tool that allows anyone to easily access and analyze this data, right? But it's also interesting to me to use that to drive my own research, right? So then you have to know what questions to ask, right? So I mean, I know some questions I'm asking are, we have these long-lived flies. We can tell what makes them live longer than control flies. So then for each thing that makes long-lived flies live a long time, I can ask, like, is there evidence that this combination of genes is also varied in humans who live a long time, right? And then you can try to piece together evidence for or against hypotheses like that from combining a bunch of different data sources. But I'm wondering, for the sorts of work that you do, are there any questions you would ask of this massive compendium of available data sets? Because we're sort of looking at automating flexible cross data set analysis, right? Like you can look at published papers, but that's each guy analyze their own data set. What we can do now, we can munch all those data sets together, and sometimes that lets you answer different questions. But I see your focus is on just analyzing the data about one person, which is of course not there in it's not there in the compendium of all the studies, because these are studies of other people who are not you, right? But But I'm still wondering if there's any questions you would be interested to pose to this sort of system.
Yeah. So the age range is something to consider too, right? Is there centenarian metabolomic data for example, right? But and there are studies
No,
that
not public. There's centenarian DNA data that is not public, but we have. We don't even have centenarian RNA, like gene expression data, let alone like methylation or something. We have DNA methylation data for 15,000 people, though, across a very wide range of ages, which is interesting, but not a lot of centenarians in there at this point.
So my interest would be, if you're able to parse out what a youthful DNA methylation or a combination of genes that are found in youth that predicts longevity, what interventions can impact that, right? Because if you win the genetic lottery, that's great. Most people didn't win the genetic lottery, right? So the big thing is on practicality. So, is there diet data? Is there track steps? Any intervention that could be potentially applied to, all right, if you have this combination of genes and you walk 5,000 steps, 8,000 steps, whatever it is, your risk of death goes down by whatever, So that'd be one way to look that I'd look at it.